Let's start today's class.
1 minute.
Ma'am, I got my PA1 bio marks.
How much did you score?
23 out of 25.
23 out of?
25.
Very good. Very good. Where did you lose the marks?
I by accident I labeled heart-shaped embryo no, mature embryo as heart-shaped embryo.
So for that, they cut 2 marks.
No, one question was that for, and the other one was for that also. I don't remember what was it. It was a 2-mark question. I lost mark in that.
Okay. Anyway, 23 out of 25 is good. Very good. Very nice. Try to not make any mistakes because you people have the potential to score full marks. I know that for every student in my class. Very good. Okay. Students, switch on your videos. Come on, quickly. Okay, what is the last thing that we have done in the previous class?
The ratio is more than more eukaryotic, example humans in there, I think.
Okay. So today we're going to see the various features. The features of the double helix was done or not?
I think it was done, ma'am, but in the recorded lecture, it wasn't there.
Okay. You weren't there in the previous class?
No, ma'am, I wasn't there.
Okay. So, anyone who was there in the previous class, have I completed the features of double helix? Ikra, were you there in the previous class?
Yes, ma'am. I just,
So what is the last thing that you have written down?
ma'am, it is the ratio is more than 1 in eukaryotes. In humans, 9.5.
Okay, okay. All right. So today we'll see the features of the double helical structure of DNA and then so on. Okay, students, switch on your videos. Come on, quickly. Let's start today's class. Let's not waste time. Also, is there any upcoming test that you people have? No, everyone is free from your tests?
Yes.
Okay, and like practicals and everything are going on in your school? Okay. All right. Okay, Ikra, Khadija, Tamim, please switch on your videos. Come on, quickly, students.
Yes, ma'am, 1 second.
Okay, okay. All right, so we had discussed about the Erwin Chargaff's rule in the previous class, wherein we said that this, according to this rule, first of all, this is applicable on the double-stranded DNA molecule, right? And secondly, after that, this rule says that all the purines will always be equal to the number of pyrimidines that are there. Now, the purines of the DNA are adenine, guanine, and the pyrimidines of the DNA are thymine and cytosine. And what is the bond that is present in between a purine and a pyrimidine, students? Which kind of bond is present in between them? Which bond is present between A and T and G and C?
Hydrogen bonds?
Hydrogen bonds are there. Now, in between the adenine and thymine, there will always be the presence of 2 hydrogen bonds. And in between guanine and cytosine, there will always be the presence of 3 hydrogen bonds. Now, we saw that the ratio of the purine divided by the pyrimidine is 1, meaning that it is constant. Now, if we talk about the ratio of this particular thing in the case of prokaryotic organism, meaning those organisms that do not have a membrane-bound nucleus present in their body. So for them, such as in the E. coli, the ratio is 0.92. Whereas in the eukaryotic organisms, such as the humans, the ratio is 1.52. Now, we are going to see the features of the double helical structure of DNA. But before we discuss this, can you tell me the names of the person who gave this double helical structure of DNA? Who remembers it? Ikra, do you remember? Who gave the double helical structure of DNA? No one remembers. No one is studying molecular basis.
James Watson, I think, and another guy, I don't remember his name.
James Watson and Francis Crick. You can also say that Watson and Crick gave the, no one asks you the first name, mostly it is the Watson and Crick. So they gave the double helical structure of the DNA. They said that DNA is present in the form of a double helix. Now let us see what are the features of this particular structure of DNA. The first is that the DNA consists of two polynucleotide chains. So one DNA molecule is made up of two polynucleotide chains, and these two chains, they are running in an anti-parallel fashion, meaning they have a polarity. One runs from the 5' to 3', and the other would run opposite, meaning from 3' to 5'. So this is the antipolarity, which is a property of the DNA molecule. Now we also know that the backbone of the DNA is formed by the sugar and the phosphate group. Now, okay, students, tell me one thing. Whenever a nitrogenous base forms a bond with a pentose sugar, what is that structure known as? A nitrogenous base plus pentose sugar is equal to what?
Ma'am, nucleoside.
Very good. Nucleoside. And then a nucleoside plus a phosphate group is known as
Nucleotide.
nucleotide. Very good. Okay, now the bond which is present in a nucleoside is known as, which bond forms the nucleoside? Also, one more thing, students, for your upcoming test that is like going to be held here in Learnivio, you have the chapter that is Principle of Inheritance, the whole chapter. And then from this particular chapter, you have the DNA, the structure of DNA, and the basic features, everything that is like from the introductory part up till the double helix model. Okay, everything from that will be coming. So these nucleoside, nucleotide, whatever you've written down in your notebooks, it will come. Now pay attention. The bond which is present to make a nucleoside, it is the N-glycosidic linkage, N-glycosidic bond. And the bond which is there for the formation of nucleotide, that is phosphoester bond. Now, if a dinucleotide is being formed, meaning two nucleotides are coming together and forming a bond. So tell me which bond will be there in between them? No one is studying this chapter. Huh? You haven't gotten the time to study, or you don't want to study. What is the reason? That no one is answering any question? Tell me one by one. Give your reasons. I want to listen to the reasons.
Didn't have time to study.
Don't have time to study. Okay. Who else? Ikra, what is your reason? At least she gave one answer though. But yes, tell me, what is your reason? Hmm? See, students, no matter if you're getting time to study or not, you are supposed to study. Even if you're not getting any time to study, I take your classes on Saturday and Sunday, right? So at least study before my classes. Okay? Hmm? Then? All right. So whenever two nucleotides are joining together, the bond in between them will be a phosphodiester bond. Okay. And talking about DNA, what kind of biomolecule is DNA? Can someone tell me?
What were the
This,
repeat again?
yeah, what, like if you talk about a DNA in terms of its like how is it made, then what would you call it? Polynucleotide. It's written right there in front of you. Okay, so DNA is a polynucleotide. Okay. You people don't like the chapter? None of you do don't like this chapter?
50-50. 50-50.
50-50. Harija, you don't like this chapter? No. Ikra, what about you?
Ma'am, a little bit.
A little bit. Okay. No matter whether if it is 50-50, whether you don't like the chapter or whether it is little bit, you have to study. This is a very, very important chapter, students. It is so important that this particular chapter is asked in the competitive exams, not just NEET, but also in higher level exams. This chapter is that important and it is the most basic. Right now, the topics that are there in this particular chapter, they are very basic. Okay, so please pay attention and try to learn side by side. Now, talking about the third feature of the DNA double helix is that the chains, the polynucleotide chains, they are anti-parallel, meaning one of them would be running from 5' to 3' position and the other would be running from 3' to 5' position. Now, the nitrogenous bases, they are paired with the help of hydrogen bonds. We know that adenine will always pair up with thymine and guanine will always pair up with cytosine. What is that one difference which is there in the case of RNA? What is so unique about RNA? Hmm?
Because the additional OH group is present at the second
Yeah,
chain.
very good. Yes, second carbon. Very good. So in RNA, there is an extra hydroxyl group present at the 2' carbon. Apart from that, instead of thymine, RNA has which nitrogenous base? Instead of thymine, DNA has thymine. So RNA has which nitrogenous base? Tamim, do you know the answer? Hmm? Do you people remember uracil? Uracil is present instead of thymine in RNA. Okay. Now, the chain, the polynucleotide chain in the DNA, they are present in a right-handed fashion, meaning the coil is present in the right-handed fashion like this. The pitch of the DNA helix, pitch means one complete turn of the DNA is of 3.4 nanometer. Now, if you talk about how much 1 nanometer is in terms of meter, so 1 nanometer is 10 to the power minus 9 meter. Now, in one turn, meaning in one pitch of the DNA helix, there are 10 base pairs present in it. Also, the distance between the base pair is 0.34 nanometer. So these are some of the things that we know with the help of which we can actually find out the length of the DNA. So whenever someone asks you to find out the length of the DNA, the number of base pairs is required and you already know the distance which is present in between two base pairs, that is 0.34 nanometer. So when you will multiply that, you will get the length of the DNA. So are all the features of DNA double helix clear to every single person here? Any doubts? Sure? Okay. Nodding your head just so that ma'am can go on. Like, ma'am, go on. Say more. Is that the case? Or did you people actually understood? Clear to everyone, right? All right. Moving forward, we're going to talk about the central dogma of molecular biology. Again, very important. Now, what is this central dogma? It was given again by the Watson and Crick. And they say that in terms of the flow of information, the flow of information is unidirectional, meaning it starts from here, it goes here. It is unidirectional. So there is a unidirectional flow of information from the master DNA to the copy DNA. Let us see how that happens. This is how you represent the central dogma. DNA is there, then RNA is there, and then protein is there. So the information travels in this way from DNA to RNA, from RNA to protein. Now, using DNA can form another DNA. So DNA can do DNA replication, because of which a new DNA molecule will be formed. Now, once the DNA is formed, this DNA can undergo a process which is known as transcription. And by that process, the DNA gets converted into RNA. Now, this RNA molecule can undergo the process of translation and form the protein. So, from DNA to the RNA to the protein, this unidirectional flow of information is known as central dogma. Is this clear? Any doubts?
No,
Also, also remember that DNA has two polynucleotide chains, right? This is one and this is the next one. Now see, during the process of DNA replication, these chains, they uncoil. Okay, for example, like a part of them uncoils like this. Like this. It would uncoil from one region, and this is, if this is 1 and this is 2, so a new DNA will be synthesized against 1 and a new DNA will be synthesized against 2 also. So you are seeing that if you talk about a new DNA, you will see that one of the strand is of the old DNA and one is a new DNA strand. Because of this, because of the presence of an old strand and a new strand, the DNA replication is known as semi-conservative DNA replication. Semi-conservative, why? Because an old strand is always present in a, like with a new strand. Clear? And then
Ma'am,
that,
will it detach from the old strand?
no, it keeps on forming and forming, going on and on. Okay? And this is known as DNA replication. Now see, if we talk about the viruses, you people must remember that the viruses, they have RNA as their genetic material, right? So now what happens in their case? So in them, what happens? Viruses, they have RNA and they can form DNA from their RNA with the help of an enzyme that is known as reverse transcriptase enzyme. So there is an enzyme in the case of viruses that is known as reverse transcriptase. And with the help of this, the viruses can form DNA from RNA. Clear? So new DNA molecules can be formed. Now, with this being said, there is another concept which is known as reverse central dogma. Remember that the central dogma is universal. It happens in every organism, whether it is human, no matter whether it is an animal or what. But if we talk about the viruses, reverse central dogma, it is an exception to the central dogma. The normal central dogma which we have studied, the reverse central dogma is an exception to that. Now what happens? This is reported by Temin in Baltimore. Just like how the central dogma was given by Watson and Crick, the reverse central dogma was reported by Temin and Baltimore. And because they discovered it, it is also known as Teminism. Now, what happened? They discovered the presence of an enzyme which is known as reverse transcriptase. And this enzyme is found in viruses. Now, viruses produces this particular enzyme, and with the help of the enzyme reverse transcriptase, the virus RNA can turn or synthesize DNA. Okay, so if you see here, what is the difference? The DNA can be synthesized, which is known as replication, but DNA to RNA, meaning the process of formation of RNA from DNA, is known as transcription normally. But in the case of viruses, a reverse reaction can happen, that is reverse transcriptase. They can help the RNA to form DNA. So this is known as reverse central dogma. Any doubts? Central dogma and reverse central dogma are clear to everyone?
Yes,
All
ma'am.
right.
Yes,
Very
ma'am.
good. Now moving forward, we will see how the packaging of the DNA is done. The DNA helix, how it is packed. Now we know that the distance between the two base pair is 0.34 nanometer. And the length of the DNA, if we try to find out the length of the DNA, so human DNA has 6.6 into 10 to the power 9 base pairs present. The number of base pair is this much. And the distance between the two base pair is this much. So if you multiply it in totality, you will find out that the length of the diploid cell human DNA is 2.2 meter. Have you people seen the dupatta of your mothers? The dupatta, the chunni, right? Uh, do you remember the length of the dupatta? How long it is, right? It's really long, right? Girls wear it, mothers wear it also. So remember that length. It is more than 2 meters. And same goes for the length of DNA. I'm trying to, you can say, relate the length so that you people can imagine how long the DNA is. Try to recall dupatta, not sari, dupatta. Okay, so when you do that, you like realize or you find out that the DNA is a really, really long molecule. But if you look at the size of the nucleus is 10 to the power minus 6 meter. It is tiny. It is really tiny. So how does the 2.2 meter long fit inside the 10 to the power minus 6 meter small nucleus? So in order to fit it, the DNA is packed. The proper packaging of DNA is done so as to make it into a smaller size so that it can fit inside the nucleus. Clear? Any doubts? Now this packaging, it happens differently in prokaryotes and then differently in eukaryotes. Now because of the very reason, because the prokaryotes have a different nuclear area and so does the eukaryotes. Now if we talk about the prokaryotic organism, such as the E. coli, the DNA is scattered throughout the cell. Now who can tell me why? Why is the DNA scattered throughout the cell in the case of prokaryotes or E. coli?
There is no cell envelope.
Cell and envelope is there in prokaryotes. You are close. Be specific. Nuclear envelope is not there. Cell envelope will be there. The nuclear envelope is not there. That is why the DNA which is present inside the nucleus, since the envelope is absent, so it is scattered in the cell. Now, remember that normally the DNA is negatively charged. Okay, normally. Now, the DNA is held with some proteins that are positively charged in a region which is known as nucleoid. The DNA in nucleoid is organized in large loops held by the proteins. So the DNA is wrapped around proteins that are positively charged and then it is present in the prokaryotic cell. This is how the packaging of DNA is done in the case of prokaryotic organisms. Now, when we talk about the eukaryotic organism, so since the complexity increases, since the size of the organism also increases, so the packaging also becomes more and more complex. Now we know that in the prokaryotic organism, there is the nuclear envelope present. Now here, the positively charged proteins are known as histone proteins. They are basic proteins. Basic. You people know what basic means? What is the meaning of basic?
Normal.
No, not that basic. I knew it. I knew you people are thinking, oh, basic, very plain. No, what is the meaning of basic? In terms of science, what is the meaning of basic? Basic means it's positively charged. Acidic,
Ma'am,
basic, you
can
people remember? Yes, Ikra? What did you say?
I have moved on.
See, remember that the proteins mostly they carry positive charge, and the proteins, the histone proteins, they are basic proteins. Now, why are they basic proteins? Because they are rich in basic amino acids. Now, you people, if you remember biomolecules, then you may recall that proteins are made up of amino acids. Now, there are different kinds of amino acids present. So, some of the amino acids are basic amino acids, such as lysine and arginine. So the histone proteins that are present in the eukaryotic organism, they are the basic like proteins, and they are made up of basic amino acids. And always remember that since they are basic, they carry positive charge. Clear? Any doubts up until now? All right. Now, the histones, they are organized to form a unit of 8 molecules. Okay, they form a unit of 8 molecules, and that is known as histone octamer. Wait. So what happens is that they like 8 of the histone proteins, they come together to form a structure which is known as histone octamer. Now, the negatively charged DNA, always remember that the DNA will be negatively charged. So, these negatively charged DNA, they wrap around the positively charged histone octamer, and then they form the nucleosome. So, once the DNA is wrapping around it, a structure which is known as nucleosome, that is formed. Now, this nucleosome, they contain 200 of the base pairs. You remember base pairs? 200 base pairs are present in the nucleosome. So, initially, histone proteins we discovered, and we talked about histone proteins. Now, the histone proteins form the histone octamer, and then the negatively charged DNA wrapped around the positively charged histone protein. Why? Because negative and positive will always attract, isn't it? Unlike charges attract. Now, from the nucleosome, chromatin is formed, and these are the thread-like stain bodies in the nucleus. So more and more packaging will form the chromatin, and they appear like beads on string under the microscope. So initially, histone is there. Histone forms histone octamer. Histone octamer and the DNA wraps around it and forms the nucleosome. From the nucleosome, chromatin is formed, and lastly, there is the chromosome. Any doubts up until now? The packaging in eukaryotes is clear to everyone? Sure?
Yes, ma'am.
All right. Now, the packaging of the chromatin at higher level, it requires additional set of proteins which are known as non-histone chromosomal protein. These are the non-histone proteins. Okay. Now, we will talk more about chromatin, but first you people note down till here. Okay. Any doubts up until now, students? Anything you people want to ask? Okay, good. I hope that you're understanding. All right, now you people can note down. Yeah, you can switch off your videos and write comfortably.
Done, ma'am.
Everyone done?
Yes, ma'am.
Done, ma'am.
Done, ma'am. Done, ma'am.
Done, ma'am.
Everyone's done?
No, ma'am.
No, ma'am.
Okay.
Done,
Ma'am, 1 minute. Ma'am, done. 1 minute.
1 minute now. Done, ma'am. Done, ma'am. Done, ma'am.
Done, everyone?
Yes, ma'am.
Yes, ma'am.
Done, students?
Yes, ma'am. Done, ma'am.
Everyone's done?
Yes.
Okay, everyone's done?
Yes, ma'am.
All right. Okay. Now, see, we talked about the chromatin. Now, depending upon the or the type, or you can say the packaging, we'll see there are two different types of chromatin. There is euchromatin and then there is heterochromatin. Now, talking about the euchromatin, so it is the region when the chromatin they are loosely packed. Okay, when they are not very close together, when they are loosely packed, that is what we call as the euchromatin. And when they are tightly packed together, we call them as the heterochromatin. Now, euchromatin regions, since they are loosely packed, so they stain very lightly because the particles in them are scarce. But if you talk about the heterochromatin region, so they are very tightly packed, so they stain very darkly because the molecules in them, they are not separate, or you can say they are very tightly packed. Now, the euchromatin region is said to be transcriptionally active, meaning it will perform transcription very easily. And talking about the heterochromatin region, it is transcriptionally inactive. So even though it stains better, it looks better, and it is present very close to each other, still it will not do any transcription. But talking about the euchromatin, it will be transcriptionally active. See, this is how different the heterochromatin and the euchromatin looks like. See, they are all tightly packed, so we'll call it as the heterochromatin. And in the euchromatin, you see that how they are separately or loosely arranged. So that is euchromatin region. Now moving forward, see, this is how the packaging of DNA molecule happens. First, see, this is the chromosome molecule, right? If we talk about the DNA, starting from here, if we start uncoiling it, so it will be histones, and then these histones will come together. They will form the histone octamer, and the DNA will wrap around it and form the nucleosome. Now, slowly, the nucleosome will start coiling and form the chromatin, and then the chromatin will start coiling and supercoiling will happen and will form the chromosomes at the end. Now, one chromosome which is formed at the end, it is made up of two chromatins, chromatids. So these are the chromatids. Chromatids are the singular parts of the chromosome which form the chromosome. Any doubts up until now, students?
No, ma'am.
Now talking about the search for genetic material. Now slowly, when all of these structures and everything was like we have studied about it, but people did not know that DNA was the genetic material. So to find out what is the genetic material in human beings, the searched, like people started to search on it. People started to form theories to study it, so as to like find out what is actually the genetic material which is in our body. So the first search for the genetic material, that whole entire thing is known as the transforming principle. This was given by Frederick Griffith. We'll discuss more about it, but first you people note these things down. The chromatin part.
Done, ma'am.